with us, so I think that's good enough. Okay, hey Ritika, thanks for joining us today. It's a great pleasure to have you. Everybody who doesn't know, Ritika is a PhD student at the University of Glasgow. She's studying cancer sciences, and
Hmm.
her research is funded by Cancer Research UK. I hope I got
Yep.
that right. She is in
Hmm.
her third year of doing a PhD,
First year.
and first year of doing a PhD. She's
Uh-huh.
in her first year of doing a you got me there. First year of doing a PhD, and she finished her master's degree from the University College London.
Hmm.
Like, so there's that. And yes, and I've known her for some time. She was a student along with me at Ashoka University. She studied biology there. After
Hmm.
which she went to the UK to complete her master's degree, and now she's doing a PhD. So yeah, so let's just start off by like maybe like telling us where you are. Where are you at the moment? Or what are you up to?
Okay, lovely introduction. Thank you so much for that. So at the moment, I am in Glasgow in my flat because I thought I'd work from home today. Otherwise, I would have had to sit on the stairs and talk to all of you because we have a shared working space. But I am in my flat and I've been here for, not in the flat, but in Glasgow for like 5 months. Before that, as you said, I was in London for over a year. And before that, it was at Ashoka where I did biology and a bit of philosophy as well.
Oh, sweet. What did you study in London in your master's degree?
Sorry?
What did you study in your master's degree in London?
Oh, funnily enough, it was just called an MSc in cancer. So it was just a year of studying cancer immunology, things like treatment, clinical trials, catching up on where we are currently, because there seems to be like it's a budding field. It has been for a while, but there
Hmm.
aren't many big breakthroughs. So it's just like catching up on research, and seeing how to go about things once we have new technology rolling in.
Yeah, because I'd imagine like there's a bunch of resources must have been going into cancer. I'd imagine like everybody's trying to find a cure or everybody's trying to figure out how to diagnose it properly. So I bet there's a lot of like research out there. Is
There
there?
is loads. There's also like with time, what's also happening is all of our machines are getting better at what they're doing. And we're exploring new avenues, which is somewhat related to what I'm doing for my PhD as well. So I didn't even know this was an avenue until I started doing my master's project.
Mhm.
What I'm using is I think I spoke a little bit about it when I took over the Instagram handle. So
Mhm.
what I'm doing right now is something called spatial transcriptomics. Big word, but it basically means that you can look at cells within the tissue where they are and then profile them, which is something we've never been able to do. So, for example, the only way to look at things earlier was say you've got an orange and you would like to see what it consists of or what it tastes of, blah, blah, blah. So, for example, DNA, RNA, protein in it, things like that. Any questions you've had to ask, the only way to do it would be to put it in a blender and get juice out of it. So you wouldn't really know where everything is. Now, what you can do is you can see where each little piece is, where you've got the seeds, how it separates, and what it tastes like, which is quite interesting. So you can just basically cut up things and make them look really beautiful.
That's very interesting. And is this like sort of like is this research towards diagnosing cancer or is this towards like treating cancer? Is that not a
Right.
difference? Like,
So what I'm doing is because pancreatic cancer is a really lethal one. The one that I'm working on PDAC, or pancreatic ductal adenocarcinoma. And
Mhm.
what I'm trying to do is I'm just trying to see how it looks like spatially, because like I said, we don't really know what spatially things look like, right?
Hmm.
I'm personally looking at, since I have a little bit of an immunologist background, I'm trying to look at immune cells within the tumor itself. So it's really fun to look at the interaction as it's happening, cell by cell within pretty colors like I've been putting up. The idea is to explore what it looks like so we can better treatment. So I would say it's more treatment based than diagnostic. But there
Hmm.
are loads of diagnostic labs as well.
Okay, okay.
Mhm.
And so this is the first time where you are able to sort of put a cancer cell within this sort of machine and look at the cancer cell. Am I getting that right?
Yes. So these machines have probably rolled out like a couple of years ago. Maybe 2019 was when they began rolling out for the public,
Hmm.
and 2019 wasn't that long ago, right?
Yeah.
So we don't have many labs working with these machines. They're really fancy, they're really huge, they take up loads of times.
Mhm.
But that also means that we have to come in at odd hours because it takes like 10 hours, 12 hours to run. Um, but yes, these spatial profiling machines is what they're called. So you spatially profile the tumor. We basically cut up really cute circles from tumors that my supervisor biopsies.
Uh-huh.
And put them through this machine, they light up in pretty colors, and then we try and see which cell is friends with which cell.
Oh, that's pretty sweet. Are these cancer cells like alive when they're going in the machine? Like, how do you keep them alive, or are they not alive?
So we don't really have to worry about them being alive or not.
Mhm.
But definitely they're not dead, because once a cancer cell dies, it gets evacuated by your body. Your body's just going to clear it out. But this is definitely from a biopsy, so it's fresh tissue when you pick it up, and then you preserve it. And these are preserved into something called FFPEs, or fresh frozen tissues. Basically means you put them through wax, put them through paraffin and formaldehyde, and you save them, and you can keep them for years and years on end. Like, you have archives that go back maybe 15, 20 years. That's what I've seen. Maybe even older than that, because I've heard people take out DNA from like 1950s as well, which we've been very scared about, by the way, because taking up a sample from 1950s, you don't know what's going to happen in the lab. You don't know what you're going to unearth. So really careful, but yes.
Oh, really? Well, what's gonna happen in the lab if you take out something from 1950s?
You never really know how it was prepared, because preparation techniques in the 1950s wouldn't possibly be as good or as careful. So you have to take, how there's safety levels in labs. So we had to take a lot of safety precautions just in case something much like the virus that's been here around for 3 years happened. We just had to be really careful,
Yeah.
because maybe there's something in there that shouldn't be in there. But that was an interesting week.
That's pretty cool. Yeah, I mean, and they do say that it happened in a lab, so who knows,
Yeah.
right?
So
Yeah.
it's not going to be us, please.
Okay, yeah. I hope they don't say this about our samples like 100 years later, and they say, "Oh no, this was in."
I hope so, because what we've done? We've really worked around our preparation techniques. I think that's something that the scientific community is now fairly okay with. I'm going to trust them with that.
Okay, I bet that's what the scientists in 19 said. But
probably,
okay.
I am walking into a trap. If this comes up 100 years later, you can't hold me accountable. I think I'm fine.
So could you tell us a bit more, about your research, maybe? Like, is there an end goal to doing a PhD research? Is there a thesis that you pick out and you pick and you say, hey, I'm going to prove or disprove this? So how does that kind of work? Like, what is the goal that you're set towards?
Right. So how it happens is, usually in the UK, you already have projects that have been mapped out, usually 80% of the time. And you try and look for funding, because otherwise it's going to be too expensive if you try and self-fund yourself. But there are already projects mapped out with end goals, with steps, blah, blah, blah. Of course, as any research goes, you're going to have to every 2 months adapt your design to how it's going to work, timelines, what you're really looking at. Perhaps you're going to unearth something you didn't expect to find. So it begins as a solid project, yes, but as time rolls on, you end up changing it a bit. What you can also do is you can propose projects to potential supervisors when you're applying for a PhD. That can be done, but then you're going to have to sit through funding competitions, because, like I said, it's going to be too hard to find funding by yourself. I think the end goal of it, in hindsight, is really small. What you're going to find out is, so for example, like I said, I'm going to look at cell-cell interaction, say, for example. Maybe I'm going to find out something so small that I say, okay, when cell 1 and 2 are near cell 3, this usually happens. That's the end of my PhD. But that doesn't really change a lot in the field until 50 years from now. Right?
Yeah.
When this will help another paper, and that's going to help another paper and another paper. And then eventually we just end up finding out loads about it. But the findings we find are so small that it often feels like you're working towards nothing, which is, I think, a problem that a lot of cancer researchers face. Because I think until a while back, we were finding, big explorations in the field. Now they're so small that it feels like you're almost chipping away at a mountain with like a spoon. So you really have to take a step back and realize what you're doing counts. It's just you're not going to be able to see it directly. So yes, there is an end goal. What I'm looking at is, like I said, immune cells within the cancer. So a couple of years ago, in the Nature journal, if anybody keeps up with it, we had a beautiful cover of it with loads of pipeline-esque structures running through tumors. And it's not just pancreatic tumor, it's lung tumors. I know breast has it, prostate has it. So these pipelines usually would be unseen, but these spatial profiling techniques, they helped us realize that it's almost like you have loads of immune cells running through the entire tumor in a very systematic way. Now the idea is if you put in any medicine through it, you've basically perfected the idea of putting medicine through the entire cancer, right? You've found a system that works, because the whole point is that the delivery of the medicine is tough within the cancer, right? Which is how chemotherapy happens. You try and fight the cancer before the cancer is able to fight your body. So the idea is to look at what these pipelines look like. They're called tertiary lymphoid structures. They just have lymphoid cells in them, and the idea is to arm them better, because they're anyways trying to kill the cancer cells anyway. So we're just trying to. It's like you're giving the police better guns. That's what we're trying to do.
Oh, that's pretty cool. Because what I heard was that our body is unable to recognize what a cancer cell is because it looks very similar to what our cells look like. So, like, what I thought, like, what ends up happening is that our body attacks the. Our body doesn't know to attack the cancer
Right.
cell.
So the idea is that it's imagine it like a race. So your cancer cells are really smart, and they're trying to race your immune cells. And there are hallmarks to cancer. So there are 7 hallmarks of cancer. One of them is evading the immune surveillance. And I said they're really smart. So what they figure out is a way to not get recognized by your immune cells. So immune cells will recognize a cell as non-self, right? When it figures out a couple of things on the surface of the cancer cell. What the cancer cell ends up doing is it ends up evading the immune cell by quite literally evading, like it just exits the space. By the time that this immune cell is going to figure out if there are these proteins on the surface or not, it's left the chat. It's gone. It's figured out a really smart way of just hiding in plain sight. And once it does that, and once it gathers a gang, which is once it multiplies a bit, it becomes really confident, and then it acts almost beautifully. I know it's very wrong to say, but there is great division of labor to the point where immune cells are going to try now really hard to break up the gang. Which is why, like I said, these pipelines that are running through it are really, they could be groundbreaking. Because the minute you realize that you now have a system of basically infiltrating this entire tumor, you have a way in, and a way that already exists. So, cancer cells they evade, but they do get recognized. Yeah, they do. And your body does end up killing some of them by itself. It's just medicine really helps accelerate the process. In fact, funnily enough, even in really healthy people, there's going to be some cancer cells. They just aren't big enough to cause cancer. In fact, you have loads of mutations as well. You have loads of mutated cells. It's just luckily enough the mutations don't end up in cancer.
Oh, that's really interesting. And the work sounds like it's not like chipping away with a spoon. It sounds like oh, this is it. This is we found where to put the medicine, and now we're gonna put the medicine there. It sounds really like groundbreaking.
Sounds very promising. But day to day in lab, once you realize you're doing really small things, and once you see your results, which are often inconclusive unless you do them 50, 100 times, it seems really small. So you, like I said, you have to take a step back, realize what you're doing, and that it does make a difference, and then move forward.
It does. I'm sure it does. This sounds amazing. The fact that you're doing it is amazing. Could you tell us maybe more about so this is a group of people that are trying to figure out this sort of delivery system of medicine, right? And it's not like your thesis, it's everybody's thesis together.
No, it is my thesis. I've got a bunch of other people in the lab, and they're working on different things. We've also got so we're a pancreatic and colorectal cancer lab. So we've got 2 surgeons in the lab, one of whom is working on IPMN. The full form of IPMN is going to be really big. I'm going to Google it so I don't mess it up. It's also a type of pancreatic cancer. Like I said, I was working on PDAC. IPMN is intraductal papillary mucinous neoplasm. So we're all working on different cancers, but mainly in the pancreas and the gut. And we've all got different projects. We do, of course, help each other out because, it's not a one-man job. But so this project is something that I'm going to be, let's not say heading, but being in charge of, deciding which direction it goes in, how to best go about it. Of course, with the help of my supervisor, with the help of my seniors, with the help of anybody around. Because basically when you bounce off ideas, you're going to end up picking it up from a load of people. And
Yeah.
when you publish research, your entire lab gets on it. So the project is mine, but everybody helps out.
Okay, well, that's pretty cool stuff. Does anyone in the audience want to kind of come in at this point and ask a question or two? Pooja or Memory? Ritika? Oh, Ritika is here. We are another student.
I've got
Bhumika?
loads of questions for myself. That is true.
Hi, Ritika. How are you?
Hello, I'm
good to-
good. Thank you so much. How's your day been?
It's been well since I'm bed resting right now. So I was hearing you out, and it's good to know about something which I'm completely unaware about. And hats off to you that you've made it sound so simple. You've been able to communicate it thoroughly. Thank you for that. So I just had one question because I had someone in my family who got the uterus cancer, and she died 7 years ago. So why is it that in the cancer, like whoever has the cancer, in the first, it's often like when she started complaining about certain things in her body, she saw the doctors, she went to different the best doctors, and in the first stage, second stage, they weren't able to detect it, and at the last stage, they were all of a sudden, then the reports come in and they say, now you're going to die. And what is the nature of the cancer, that we're in this why are we still not being able to figure out, or to to say that, okay, now you have reached at this stage, maybe we can cure you. But most of the times what happens is like you reach at the last stage and then you have nothing, you just die out of pain. So why is it that, what nature makes it so difficult for the researchers to reach to that point and to save life of people?
Right. Firstly, I'm really sorry to hear about that your family member had to go through that, and then your loss as well. I think the problem really comes from not diagnostics. I think diagnostically we're doing okay. I think the problem comes with execution at ground level, especially with things like breast cancer, prostate cancer, uterine cancer, ovarian cancer, things that are usually too shameful to talk about to family members. So I think this is a problem I'm certainly sure if happens in breast cancer. Wouldn't know about uterine as as certainly, but for example, in India, we've got loads of you'd think we've got loads of research going on and everything, but we do know we have a shortage of doctors. Right. We do know that we have a shortage of funding. We have a shortage of cancer centers. We also are really short on awareness. So for a woman to stand up and say, my breast hurts, it's going to take a while for her to reach out to a head of the family or go to a doctor. It's not as easy as just taking an Uber and heading to a hospital, right? For most people, really, it's going to be a 2-hour drive to a state or a city they don't know, an underfunded one probably, where the doctors aren't going to be oncologists. You don't have oncologists everywhere unless it's a special cancer center. Right. And opening these centers is really, really hard right now because India, and I'm not sure about Nepal, we're really struck in palliative care, which is like, you said, in the later stages, management of pain. So we really fall short on, finding it early on. This is something that perhaps the West is able to now catch on to, and it's taken them a while to reach here as well. But even with breast cancer, women usually come in with 3rd or 4th stage disease. In India, more than 50% of them, which is really bad because that often means that you don't just, get away with surgery. You have surgery, radio, chemo, and all of these things take a toll on you. And things like radio and chemo, they end up, affecting the body in such a way that even that causes pain. So it's not just the cancer that's hurting you, it's the medicine as well. It's just the idea is that this medicine is going to kill the cancer before it starts killing you. It's just a fight between your body, the medicine that goes in it, and the cells in your body that aren't your body anymore, which are cancer cells. But I think it just ultimately falls down to execution and how we end up dealing with it as a country. I think everywhere in the world right now, and that's something that COVID has really unveiled, is that we have a shortage of doctors, we have a shortage of equipment, we have a shortage of resources, and that ends up showing when it comes to one-on-one care. So even though research would think that they've made a jump there, so for example, research wouldn't be worried about diagnosing uterine cancer. They would know we've got good diagnostic tests. We can always make them better. We can make the machines better, etc. But we've reached there. It's just executing that is going to be a big issue unless it gets funded by policymakers. And remember, policymakers aren't usually researchers. They're people who often have no idea about cancer or hospitals or doctors, or even don't have the same motivation as some of us. So that really ends up showing.
Okay. So it's not the it's not the technical aspect that's hindering the diagnostic process, but it's the lack of communication between two different sectors of people and coming together. Okay.
That's what it often comes down to. Unless it was a, unless it's a rare cancer, in which case, it takes a while for even the diagnostic lab to come around and see what it is. Like, there are some cancers that are maybe only 16 people in the world have. So then in that case, it's going to take a while for that, to even show up in testing or for them to know what they're looking for. But most cancers, I would say diagnostically are doing better. Of course, they have poor prognosis, which means that they're often found at later stages, like you said. So it's often too late when they're found, but loads of people working on it. I hope it gets better.
Yeah, let's hope for that too.
Mhm.
Thank you.
Thank you so much.
This is amazing stuff. Like every aspect of this is really incredible. Good to listen to. Well, anyone else? Bhumika, do you have a question? Or Memory or Pooja? Well, Ritika, why don't you tell us about how? Oh, Shashwat has a question. Shashwat, hey, go ahead.
Yeah. Hi, Ritika. It was nice to hear you and meet you. I loved your stories from Instagram on the
Thank
Kuhiro
you so
page.
much. I'm glad I could keep you
Yeah,
entertained for a while.
I had one question. Like, I was just curious. Like, why did you choose pancreatic cancer specifically? There are like so many different type of cancer. So, like, what made your interest like in pancreatic cancer specifically?
Right. So initially, funnily enough, I wasn't interested in pancreatic cancer. I don't think anybody goes and, says, oh, pancreas is where it's at. I don't think that's how it happens. I was very much interested in blood cancer, and that's because someone really close to me had blood cancer when I was a child. And seeing that something in you shifts at some point, you as a child end up having a really, really naive idea of, you know what? Nobody can figure this out. I'm going to grow up and figure this out. So I ended up working or studying about blood cancer. And in my master's as well, I was working with multiple myeloma, which is a blood or bone cancer. And after that, when I was applying for PhDs, I saw this position open. And it just so happened to be headed by like my secondary supervisor was somebody who had made the software that I was using when I was working on blood cancer. And that software is really groundbreaking. It's an open access software called QPath. So it's a pathology software, and I'm really interested in pathology. So when I realized that this was something that we're doing, I ended up looking into pancreatic cancer. And then I realized that, oh, my bad. Right. And then I realized that the project was working with PDAC, which has a 5% survival rate after 5 years. That's so terrible. That means 95 out of 100 people end up dying within 5 years of the disease being diagnosed. Right. That was a blasphemous statistic for me to come across. And I was like, okay, I'm going to look more into it. And the more that you look into it, you end up realizing that unlike gut cancer or stomach cancer or lung cancer, it's not talked of as often, but it does, like, loads of people do suffer from it, especially in Scotland. So I think it's the third most or second most common cancer. And once I realized that this was something I was interested in, its goals matched mine, and it used techniques that I had used in the past, I think I came around to pancreatic cancer. Of course, this also means that you don't have to stick to the cancer you research in. It's more about learning the skills to work around something. So for example, I use the same skills to work with blood cancer, and I could transfer them to pancreatic cancer. So it doesn't matter what you've done in your bachelor's, master's, or PhD. You can always move around labs or cancers.
Yeah, thank you very much for sharing that with us. And I'm really sorry to hear that someone close to you had to suffer from cancer. I know it's really bad.
Oh, thank you so much.
Yeah.
Touch wood, they're all well
Yeah.
now. So thanks to medicine, it does do some well.
Yeah, it does like really good if we can find a cure fast, but I don't think like it's possible at the current moment, or like in the next 50 years or so. Maybe something groundbreaking research might happen. So yeah, I'm hoping like we find a cure for cancer soon.
Right. I'm hoping so too. I think it's actually quite the term cure for cancer is very funnily looked at in the cancer research field, because there is no one cure. What's going to happen is there's going to be a probable treatment that may work. It may have a 20% chance of working, 90% chance of working, 12% chance of working. It's just It's hit and trial. That's the problem. The problem isn't that the treatment isn't sure shot. It's just cancer is such a thing that it's going to be different body to body. In fact, it's going to be different. Say, for example, cancer is metastasized from your lungs to your brain. Let's say that. It's going to look different in your lungs and different in your brains. In fact, it's going to look different within the same tumor itself. It's going to have a region that looks very different to the region it started off from. And this, what you call heterogeneity, is what makes it very tough for treatment to happen. It's also the smartness of these cancer cells that makes most people resistant to treatment. So there are times that you're going to have some improvement, it's going to fall down, some more improvement, and it falls really down. It's the cancer cells are so smart that they realize this is what you're putting in the body. I'm going to figure out a way to evade that as well. Now figure out a second treatment that works. And again, you go into hit and trial, try and figure out the entire thing. But of course, this takes a massive toll on patients. So oftentimes, by the time you reach second line or third line cancer treatments, you've lost the battle. So it's not the treatment where we're failing at, it's the heterogeneity of the cancer within a patient, between patients, between cancers, that you can't really tell what's going to happen. It's more of a find out once you've gone about it sort of a thing.
That's really interesting. I think I just want to turn every 1 minute of this video into a reel and share it, because everything is just, it's really, really mind-boggling, everything that you're saying. I just wanted to ask, so like, so what's next for you personally? Like after you finish your PhD, when does your PhD finish? Like, when do you, is it like, oh, now I've done it, see it, I've found the partial cure for something, and now I'm good to go? Or is it after you finish a certain number of years of research? And what happens after you finish your PhD? What does that look like?
Right. So technically, I will only get my degree once it has been accepted and once I've defended my thesis. That would ideally be within another 2 years, because I have been funded for 4 years, but the goal would be to finish within 3.5 years. And the idea would be then to finish this project that I'm working on. Like I said, we've got an end goal. The end goal is to characterize the cells within these pipelines, like I said, that run through pancreatic cancer. And once I've characterized them, which basically just means you've profiled them for DNA, RNA, proteins, seen how they interact. Basically, it's like stalking somebody. So once I've stalked what that neighborhood looks like, and we call them neighborhoods as well. So once I've seen what that looks like, I would then present it within the next 2 years and hopefully be done with my PhD. Let's hope. But personally, I'm not sure yet, because what's been happening in research lately, and this is, I get loads of these questions on LinkedIn as well. There's not many academic jobs available. Not in the West anyway, which means that now people are turning away from academia post PhDs and postdocs, and they're turning towards industry jobs. And industry jobs pay well, are 9 to 5s, but often end up taking away the novelty of research. So you don't get the same kick out of it. So, we come in with a passion to research, to look at things, but it often takes away that autonomy, because then you're going to be working for a company and for their goals and not your own. So personally, I'm a little bit split between the two. I would, of course, like to stay within academia, look at postdocs as well. I don't think as many people are opting for postdocs after PhDs now. I think things are changing and have been changing for the past 4 years because of funding and inflation and the economy not being so good to us, etc., etc. Even migration, a lot of rules and laws have changed around it. So after all those influences, I think I'm going to try my best to stay within academia, but should it not happen, I would be open to an industry job within cancer research itself.
But please to inform you that Kuiro Class is going to give you your own research center someday.
Oh, very lovely. I would love to have that as my primary option, and then industry jobs are going to be my backup.
Yeah, okay, we got you. So what is it about industry jobs that make it like sort of like not as cool as research job? Like I understand that the passion of like what you're trying to find out is not going to be your own passion. It's going to be like the company's passion of like, find this certain chemical or that chemical, maybe, I don't know. But is that what makes it? Do you think like companies are not, like pharmaceutical companies are not pushing for a good agenda, or?
I think they're definitely pushing for a great agenda. It just happens that they're working at a base level. So they're going to be searching something that can be very widely applied, right? That's what they would want. So for example, say they're looking at malarial cells and something within that. So I would say, for example, if I was not an immunologist but a geneticist, and I would say I'm really good at XYZ techniques and I can work within malarial research, and I take up an industry job with, say, Johnson and Johnson. Every day I'm going to be now working on something my PhD was not in. My PhD was a niche within pancreatic research. I was looking at a certain type of cells. So even when I say immune cells, maybe it'll happen that I'd look at only a certain type of B cells, right? I'm going to go really deep. I've ended up finding expertise in a place where I might not know anything else about the rest of the research, but I'm going to know loads about just one certain thing. The problem with that is I would now be an expert in that, right? But I cannot use that expertise in my job anymore. I'm now doing something that I would have learned in my bachelor's. It's just I have, with my PhD, learned how to critically think, how to head a lab, how to work around and design an experiment, etc., etc. I've learned skills, but I'm not able to use my expertise anymore. So it often, like I said, doesn't result in a lot of novel research. So it doesn't give you the same kick anymore. Of course, there's benefits such as loads of pay, such as a good 9 to 5. You have a good work-life balance, or hopefully better than an academic. But it does come sometimes at the cost of your interests. So what usually happens is people stay for a while in academia and then end up shifting to industry after a few years. I don't know how that would go for me, but this is what I've been told. This is all something that people who've joined industry jobs and then people who joined, academia positions have told me. So a bit torn about both of them.
Yeah. Now I kind of wish that you don't find your hypothesis, like conclusion soon. You just stay there for a while.
So that I can stay as a student for a while, stay in limbo.
Yeah. Do you have any last words for us, and then maybe we could open to a floor for like 2 minutes, and then we could share. Do you have anything else you want to talk about?
Anything else like a wisdom nugget? I think I am nowhere to be given out wisdom nuggets, but one thing I would say is stick with your gut when you end up choosing what to study. Stick with whatever interests you at whatever level that is, even if it's in 10th grade, 12th grade, bachelor's, master's, PhD. Just end up moving to something that really catches your eye or motivates you, because there's going to be times at every stage that you're going to run out of motivation, and the only person who can fuel you is going to be yourself. So it truly helps to be to be able to take a break and tell yourself that your ultimate passion or interest comes in from yourself, because you're never going to know everything about anything. Like even at this stage, if you were to ask me a question, there's going to be a chance I'm going to write it down and be like, I'm going to get back to you because I'm not going to know about it. Same happens with my supervisor, who is so well studied, right? The only reason that we can stay where we are is because we're curious. So I guess just staying curious really helps. It just helps get through tough days. It helps find motivation, find what you want to do. Because I don't think anybody wakes up one day and says, this is what I'm going to do. It's just you end up you end up coming across it while you're trying to figure out what you might like. So just keep figuring out things you like, and you don't have to stick to one thing. You can like loads of things and then juggle between them for as long as you can until you have to choose the one thing you want to be an expert in.
That's really amazing advice. Cool. Anybody have any questions? Any closing questions? Pooja, come on, you haven't said anything. Or memory of
I've been a very good listener, and thank you for sharing this, because I have a bad memories with the cancer thing, because 1 and 2 people of mine passed away due to the cancer within 2 years, and the research that you are doing is what one of my grandmother had. So it's kind of overwhelming to listen to everything and just think cancer all the time, and it's foggy, to have a brain fog. So it is nice that you share, and happy Women's Day, everyone. Yeah.
No, thank you so much for joining. I can understand it can be overwhelming. In fact, it does get overwhelming in the lab as well, and that's something that I think all of us face. There's going to be days that you come back feeling really defeated because you just hear about losing a patient or figuring out things that you would have expected that by the end of this week I'm gonna reach here, and you don't, and you come back really defeated, or you realize that the answer you were looking for is such a complicated answer that you have to really dumb down your question, so you end up thinking you're not going to make a mark as you thought you would. And of course, I think not just me or you or a bunch of people on this call, I think everybody ends up, knowing somebody close to them that has suffered from this. And 1 in 2 people now, at least in the UK, at some point will have cancer. So that's a statistic as big as, somebody you're talking to, one of you might be it. And with a statistic that big, it just ends up being something that's really gonna affect the coming generations. It's gonna, from what I've heard, get worse. So the idea is that, again, it's a race. I hope we reach the end line before whatever we're looking at does. But again, it's really important to I think not take it too personally. And I know that sounds like really bad advice, but that's the only way that you're going to be able to think, okay, this is my bit, I've done that, and that's the only thing I can do. It's just one of those things where once you're dealing with it, you have to of course there's going to be personal motivation. So I'm not saying be impersonal about it, because you're not going to be a good anything if you're not really personally motivated about it. But again, it really helps to not let it take power over you that way. Otherwise, it's going to be really paralyzing.
Thanks, Nara. And thanks, Pooja, for your question. Anybody else have any closing remarks? Otherwise, we'll close.
Do you have any advice for someone who's like searching for a PhD option right now?
Oh, yes. Depends really. Is this a PhD in STEM, or are we looking at what are we looking at? Because the advice is really going to vary from STEM to non-STEM.
No, actually. So I meant like PhD in the same field.
Oh, same field. Lovely. Okay. So I
Yeah,
think a good starting
biology.
point is to see what countries you'd be looking at. If you're looking at your home countries, if you're looking at international countries, and see how they fund students. So, for example, funding in the UK is very different to Canada or the US or Europe. You're going to realize that it's going to be much easier in some places. For example, I know finding funding in Canada is significantly easier than finding funding in the States. So those give you, an idea of where you want to be, which gives you an idea of what universities you should be looking at. Once you figured out that, try and look at the departments that you might be interested in. Look at supervisors, click on the web pages. It's going to take a while, but just snoop around. See what sort of a lab it is, what sort of research they do. And then often around, this time, actually, it's going to be December to May is when positions are open. So around that time, just end up seeing what research interests you really, the questions they're asking, and if that's a question that you can give 3 to 8 years of your life of, because sometimes a PhD ends up going to 8 or 9 years as well. Once you figure that out, try and send them an email, a little one saying, hello, I'm very interested. What do you do? Can we have a meeting? Short meeting, and then you move on to the application process. So I think a good place to start is country, university, department, lab, and then if it's a good fit for you or not. And then, of course, websites like, Find a PhD really help. So keep a weekly check on it if you're actively looking for PhDs, and maybe you'll come across a project that really interests you.
Thank you very much.
No worries.